Thank you.
Let's move to the second talk. Now I'm pleased to introduce our second speaker, Dr. Yuchen Lu from the Virus Manufacturing Department of ProBio. Dr. Lu holds a PhD in molecular biology from Newcastle University and is currently a member of the Manufacturing Science and Technology team at ProBio. In this role, he is responsible for optimizing viral vector manufacturing processes and ensuring their successful transfer and scale up from development to GMP manufacturing. In this talk, Dr. Lu will discuss lessons from IND clearances in CAR-T development, from LVV process development to IND. Now, I will hand it over to Dr. Lu.
Right. Hello.
Yeah, we can hear you. You can share your screen.
Can everyone see my screen just fine?
Yeah, it's good. You can start.
Good afternoon, everyone. My name is Yuchen Lu, and I'm a senior scientist working in the MSAT team.
Oh, sorry, Dr. Lu, sorry to interrupt you. We found your voice is unclear. How about, adjust it. If it doesn't work, maybe we can remove the earphone.
Okay.
Yeah. Hope that will be better.
Is that better now?
Yeah, better now. Okay.
Okay.
Yeah.
Great. I'm a senior scientist working for in the MSAT team at the Virus GMP Manufacturing Department of ProBio. Our team takes part in LVV process development and is responsible for the transfer of manufacturing processes across departments, as well as in the troubleshooting of production-related issues. So today, my presentation will cover topics including the optimization of LVV packaging systems, manufacturing process development, the choice between using adherent and suspension production systems, and the scale up of manufacturing processes. Now, firstly, I would like to go through what is the current industrial standard third generation LVV packaging system and how we got here. The second generation LVV packaging system preceding it consists of a transfer plasmid carrying the gene of interest, packaging plasmid expressing the 4 LVV proteins, and an envelope plasmid for the expression of the envelope glycoprotein. The 5' LTR of the transfer plasmid carries a weak native viral promoter that requires activation by the TAT protein expressed on the packaging plasmid. The third generation system improved on this by replacing the native viral promoter with heterologous constitutive promoters such as CMV, ISV, and EFV1 alpha that do not require TAT activation, which helps reduce the number of viral proteins needed in the system. Now, self-inactivating 3' LTR were also designed to reduce the risk of insertional mutagenesis, and the packaging proteins were also placed on 2 separate plasmids instead of all of them being packed onto one. So these changes lead to reduced risk of RCL generation during LVV production, as well as the risk of potential genotoxicity. The added complexity to the transfection process, because now we're using 4 plasmids instead of 3, means that the third gen system tend to have lower yield compared to the second gen system. However, this was outweighed by its better safety profile, making it the current industrial standard in CGT LVV production. Next up, I would like to go through some of the considerations we should take when designing our LVV packaging system. The first 2 considerations are safety related. The antibiotic ampicillin, which is commonly used in E. coli culturing, is a strong allergen whose use should be avoided in the manufacturing of biologics. The Chinese pharmacopoeia extends this ban to the use of ampicillin-resistant markers on plasmids used in the production of LVV. To meet this regulatory requirement, kanamycin-resistant genes are typically used to replace the ampicillin ones as a selection marker. Next, the LVV transfer plasmid also usually carries a WPRE sequence, which plays a key role in enhancing mRNA stability, translational efficiency, and nuclear export. However, the wild-type WPRE sequence contains an open reading frame encoding a truncated peptide of the WHX protein, which may be related to tumorigenesis. So mutating the WPRE to abrogate the translation of this peptide is necessary in ensuring the safety of LVV manufactured. Next up, the stable expression of the transgene relies on a strong promoter. Nowadays, it is usually one of these three, CMV, RSV, and EF1 alpha, all of which are universal and constitutive. However, there are also can be times when a more tissue-specific promoter is more desirable, depending on the therapeutic goal and the downstream application of the specific LVV. Some of the examples include TGB receptor for liver cells, CD11B for myeloids, and SFFB for immune cells. Choosing the wrong promoter can have a lot of negative implications, some of which include the over or under expression of the transgene. And in the case of underexpression, this can lead to a high virus copy number to CAR positive ratio, which is detrimental to the production process of CAR T. Other effects include the silencing of the transgene by epigenetic modification over time, leading to the reduced therapeutic effect. Off-target effects may also occur in some applications, and the immune system may also get triggered, leading to the clearance of the transduced cells by the patient's immune system. Envelope protein choices dictate the type of cells the LVV particle can infect. At the moment, the most prevalent LVV envelope protein is VSVG. It's got the advantage of having a broad range of tropism due to its ability to bind to the ubiquitous LDL receptor. It's got the advantage of high stability, which allows it to withstand a lot of abuse and stress during the downstream purification process. And it also packages into functional viral particles very well, which means the upstream titer that we can also tends to be very high. Now, because of its prevalence and its use over the years, it's got a lot of regulatory acceptance and a good clinical track record, as well as a well-characterized GMP manufacturing processes for it. On the other hand, it does come with quite a few limitations as well. Firstly, it has relatively low infectivity against cells with low LDL receptor expression levels, such as natural killer cells and hematopoietic stem cells. It also faces the problem of serum neutralization, which limits its use in vivo gene delivery. Its cytotoxicity also hinders the development of stable producer cell lines, often requiring the use of an inducible system, complicating the manufacturing process. The issue with autotransduction, where the newly packaged viral vector infects its packaging cell line, leading to a drop in titer over time, can also be problematic. Now, some of the alternatives to VSVG include the RD114 and BAEV glycoproteins. Now, these two envelope proteins target the neutral amino acid transporters and are thus more suitable than VSVG in infecting certain types of cells, such as NK cells and HSCs. They also have the advantage of having lower level of cytotoxicity in comparison. Our experience with these glycoproteins showed that replacing VSVG with these two typically leads to lower levels of LVV titer in upstream harvest material. However, it is important to keep in mind that the transduction titer results can change significantly based on the cell lines used to perform the assay. Now, we've got past experience working with these glycoproteins due to our clients' requests to experiment with different LVV pseudotyping, and we found that the biggest challenges in using these two instead of the SVG is to optimize the upstream process to get higher viral titer in the harvest material, while not a lot of changes needs to be done to the downstream process to achieve high recovery. At ProBio, our platform LVV packaging system is a third-generation full plasmid transfection system. Our plasmids carry kanamycin resistance genes and mutated WPIE. They're clearly traceable and are DMF ready at the US FDA. We have got stocks of GMP-grade helper plasmid that are ready to use, and all of this was designed to make sure that our platform process is robust, streamlined, and flexible for most standard LVV manufacturing projects. However, we're also very willing to make modifications to our process based on our clients' needs, such as changing the LVV pseudotype or using a stable producer cell line. Next up, I'll go through our platform LVV manufacturing process. For adherent processes, after viral thaw, we'll typically go through a few rounds of expansion in flatbed flasks and CF10s before seeding the production culturing vessel, which are usually made up of multiple CF10s connected in parallel. This is typically followed by transfection with 4 plasmids, and then harvest and clarification. Our adherent process uses a CDMO authorized 293T based cell line and can reliably achieve an upstream titer of around 5E6 to 5E7 transducing units per mil in the harvest material. Now, the adherent culturing system was the industrial standard for a long time and had built itself a good track record over the years. It also has the advantage of easily achieving consistent yield during scale up, since we're merely increasing the number of culturing units, for example, by going from using 1 CF10 for a 2-liter production to using 12 CF10s for a 24-liter scale production. Now, however, it does run into the problem of being very costly in terms of consumables, as well as being very labor intensive due to the need for media exchange and frequent cell detachment. So much so that at scales beyond 48 to 96 liters, it becomes completely cost prohibitive. Now, adherent culturing also comes with the disadvantage of having to use serums such as FBS in the growth media, which again introduces the risk of contamination by animal-originated viruses, increasing the burden of safety testing after production. On the other hand, we can use a suspension culturing process for the production of lentiviral vectors as well. So after viral thaw, the cells are typically expanded using shake flasks or small-scale bioreactors to the target volume and density, and then used to inoculate the large-scale single-use bioreactors. Again, this will be followed by transfection and later by harvest and clarification. For our suspension process, we use a proprietary 293T cell line adapted for culturing in suspension, again with FDA DMF registration. Our suspension process typically yields upstream titer of between 1E7 to 1E8 transducing units per mil. Now, compared to an adherent process, a suspension culturing has the advantage of producing higher titer in the upstream harvest material. It is highly scalable and better suited for large-scale commercial production. Another advantage is that it uses serum-free, chemically defined growth media, which is considered to be safer than ones that contain animal serums. And it also helps us reduce cost in labor and improve production efficiency. However, it also comes with its limitations. For example, culturing in suspension typically has the disadvantage of having higher contents of cell debris in the harvest material, which can add challenges to the downstream process. And in addition, achieving consistent yield during scale up also tends to be a lot more challenging when using an adherent process. Challenges encountered when scaling up suspension process mostly comes from differences across bioreactors in areas such as the scale of production, the type and specification of the impellers, the design of the culturing vessel itself, such as the height to diameter ratio, the absence or presence of baffling plates, and the difference in sparger design, and the PID algorithms for pH, DO, and temperature control. The scale-up strategies for suspension culturing typically focuses on keeping the chemical factors such as pH, DO, and partial pressure of CO2 consistent. For geometry-related physical parameters, because of the laws of physics, it is impossible to keep everything constant, meaning that we will have to prioritize some and drop some others. Typically, we'll try to keep the power per volume ratio and airflow rates consistent, while parameters such as impeller tip speed, the pumping per volume, mixing time, and superficial gas velocity have to take a backseat. And because of the wide range of variables that needs to be considered, very often the success of the scale-up operation comes down to our team's experience with different models of bioreactors and familiarities with the sensitivities and insensitivities of the cell lines we're working with. The downstream process for both adherent and suspension process tend to be very similar, and they typically consist of an initial clarification step for the removal of cell debris, followed by a concentration step to get the virus bulk down to a more workable volume, nucleus incubation for the removal of HCD and plasmid residue, and then chromatography for HCP removal. Lastly, it will be the sterile filtration and buffer exchange steps to put to ensure the sterility of the virus bulk, as well as putting them into their final formulation buffer. The exact downstream process flow and the order in which these steps are performed vary from project to project, and will often shuffle things around based on the result of our downstream optimization. For most LVV projects, our downstream process can reliably achieve a stable recovery rate of around 30%, and sometimes go to as high as 60% if the process is optimized enough and simple enough. Along with very high now, this also comes with a very high stable removal of process-related impurities such as HCP, HCD, and plasmid DNAs. Next up, I would like to go through a few things that are important to keep in mind when planning a GMP batch production. Firstly, it's the transfer and alignment of analytical methods, which is absolutely crucial, especially the methods for quantifying transduction titer. Differences in cell lines, antibodies, and experiment design can all lead to significant fluctuations in titration results, which can cause inconvenience to the downstream application of the LVV. Another aspect that is often overlooked is the amount of material that will be lost or sampled during large-scale GMP-grade production. For example, often up to several liters of upstream cell culture will be sampled in the form of unprocessed bulk, supernatant, control cells, or end-of-process cells for various types of safety testing and test qualifications, such as RCL, mycoplasma, adventitious virus testing. Up to hundreds of vials of filled product may be taken for release testing, regulatory sample retention, and stability study. The automated aseptic filling process used in GMP production can also incur losses due to things such as fluid being trapped in single-use filling assemblies, flow path, or just due to filling rejects. Now, lastly, when specifying the final filling volume, it is also important to keep in mind that if you want to make sure, for example, at least 1 ml of material can be extracted from a vial, it is probably safer to increase the filling volume to 1.1 ml due to filling inaccuracies. Yep, that is the end of my presentation today. Thank you very much for coming.
Thanks for Dr. Lu's presentation. Now let's come to the Q&A session. Yeah, once again, we warmly invite you to submit your questions in the chat or Q&A panel, and also follow us on LinkedIn and the Kakao channel for more online resources and updates. Here we received several questions. Let me paste and send in the chat box so everyone can see it. The first question is, does ProBio has experience in producing LVV using stable producer cell lines? Could you support on this question?
Yes, no problem. So yes, we do have experience in using a stable producer cell line, not for LVV, but for RVV, but then again, they're quite similar. And what we found is that using a producer cell line does indeed help increase the upstream titer by quite a bit. But then again, its application is still going to be quite limited for most of the early phase clinical trial projects because of the cost of developing a stable producer cell line, as well as the lead time it will take. So for most early stage processes, it just doesn't make too much control and logistical sense to tap into a producer cell line instead of just using transient transfection production method.
Thanks for your sharing. And the next question is, how does ProBio decide what downstream purification route to use for a specific LVV project?
So the downstream purification steps for most of the LVV purification are quite um, are quite constant. It's usually just the same handful of steps that can be shuffled around a bit. We'll typically do a few small scale runs to test out a few different purification routes and just to see which one gives the best results. But again, the exact process will depend on the project, and sometimes we can do that nucleus incubation in the bioreactor prior to harvest. Other times we'll do it on after the initial concentration step. A lot of times, nucleus incubation can lead to the formation of precipitations. So that in these cases, we'll have to add a clarification step afterwards to make sure that these precipitations do not clog our columns. And sometimes if our clients want the LVV to be extra pure, then we can also do 2 steps of chromatography instead of 1. However, this does come with the drawback of sometimes lowering the recovery of LVV. So it really depends on does our client want higher purity or do they prefer higher yield? And what is their downstream application?
Thanks for your clear explanation. And the next question is, what does the most basic GMP grade LVV manufacturing involve, and how does it compare to typical average GMP LVV manufacturing? Would you support on this question?
Sorry.
Yeah, you can find the question in the chat box. It's from Makul Gupta.
Yeah, so it kind of depends on what is the application, because I believe for early stage clinical trials, the requirement can be not as high as like a commercial scale GMP manufacturing. A lot of times it comes down to the strictness of environmental monitoring, the usage of open exposure processing steps, and sometimes some processing steps such as centrifugation, which should be avoided at commercial GMP, but may be considered acceptable at lower basic levels. Yeah, so the small things in these areas.
Thanks for your answer. And the next question from Kenneth: what transfection reagent is used in the ProBio? Does ProBio have the pass criteria for functional or physical titers?
Yes. So for these assays, when we're doing the testing, we'll have a reference sample. And a reference sample is usually should be within a predefined range that was identified during the method qualification, which is typically plus minus 30 to 40% ISD around the mean. Okay, what measures? Right, so for host cell proteins, the downstream steps that are used for its removal is usually takes place in the first step, ultrafiltration. Typically, we would use hollow fiber filter module with a molecular cutoff point of 300 to 500 kilodaltons, which can help remove a lot of the proteins during the concentration process. And then the next one would be the multimodal chromatography we use, which is basically improved gel filtration step, which can help trap a lot of the whole cell proteins in the resin. And lastly, if needed, we can also do a second anion exchange chromatography step to help remove any residual positively charged proteins that are still left at this point of the process. And typically, this is more than enough to ensure a low HCP concentration in the final product.
Thank you for your time and thank you for your answers. That's all the question from my side. Now we